Are you risking falling behind on groundbreaking research that could redefine your field? Today, we're diving deep into an urgent area of innovation you absolutely need to know about. Welcome to Peptides Today. I'm Sonia Rao, your pipeline tracker. My goal is to bridge the gap between complex lab science and actionable research insights. We're examining a critical advancement: peptide-based vaccines targeting ovarian cancer epitopes, highlighting their potential impact on future studies and discovery.
Ovarian cancer, an intractable research challenge, often diagnosed late, demanding innovative approaches for study. High recurrence and chemoresistance in preclinical models underscore an urgent research need. New therapeutic strategies are essential, moving beyond traditional cytotoxic agents in studies. Peptide vaccines offer a promising pathway to enhance precision and efficacy in the research landscape. These vaccines are designed to stimulate a targeted immune response against tumor-associated antigens. The goal is to enhance the immune system's ability to recognize and eradicate specific research targets. Immunotherapy has transformed oncology research, leveraging the body's intrinsic defense mechanisms. However, ovarian cancer studies historically show less responsiveness to checkpoint inhibitors. This limited efficacy is attributed to a complex immunosuppressive tumor microenvironment in models. Peptide vaccines could carve a unique niche by educating T cells to attack malignant cells. The concept is elegant. Identify protein fragments uniquely expressed by ovarian cancer cells, synthesize these peptides, then deliver them to elicit a robust cytotoxic T lymphocyte response. This strategy seeks to re-engineer the immune system to act as a precision-guided research tool. It's a targeted approach against tumor cells, aiming for superior specificity in laboratory settings. We must examine the broader landscape of peptide immunotherapies targeting ovarian cancer research. This analysis will focus on clinical viability, current challenges, and future outlook for such research. Our discussion is grounded in existing research and crucial regulatory considerations for discovery.
That was just a glimpse into the foundational challenges and initial promise of peptide vaccine research in ovarian cancer. The scientific community is tirelessly exploring these avenues. For more in-depth analyses like this and to stay ahead of the clinical curve in peptide development, make sure to follow me, Sonia Rao, on LinkedIn and subscribe to the Clinical Cadence Digest from Peptides Today. We'll be right back to dive into specific targets, mechanistic intricacies, and the regulatory pathway.
Several tumor-associated antigens like NY-ESO1, MUC1, WT1 are prime peptide vaccine targets. These are overexpressed in ovarian cancer cells with limited expression in healthy tissues, a research advantage. NY-ESO1, a cancer testis antigen, is extensively studied in high-grade serous ovarian cancers. Peptide vaccines targeting NY-ESO1 aim to induce both humoral and cellular immune responses. Early trials in ovarian cancer demonstrate promising immunological responses in research cohorts. Evidence of preclinical activity, such as stable disease, is observed in heavily pretreated research models. MUC1, another glycoprotein, is a focus for peptide vaccine research against aberrant forms. Studies involving MUC1 peptides show safety and antigen-specific T-cell responses in certain models. The mechanism of action centers on antigen presentation by antigen-presenting cells, APCs. Within APCs, peptides are processed and presented on major histocompatibility complex molecules. This presentation activates CD8+ cytotoxic T lymphocytes and CD4+ helper T cells, crucial for research. Activated CD8+ T cells then migrate to the research target site, initiating cellular destruction. Peptide vaccines offer high specificity. Unlike nonspecific cytotoxic agents used in traditional studies, they are potentially combinable with other immunotherapies or conventional research treatments for synergy. The primary research challenge is achieving sustained anti-tumor immunity in immunosuppressive microenvironments. Future research must incorporate novel adjuvants, delivery systems, or combination strategies to advance. The regulatory pathway for peptide vaccines is complex and lengthy, requiring extensive data for approval.
The prospect of peptide-based vaccines for ovarian cancer holds significant promise, reflecting a strategic pivot towards highly targeted, immune-mediated therapies in research. We anticipate continued advancements in identifying optimal peptide antigens and improving vaccine delivery methods. The long-term outlook for peptide vaccines in ovarian cancer research is cautiously optimistic. By priming the immune system, these vaccines could render research models more susceptible to current therapies, leading to more durable responses. Stay ahead of the clinical curve by following me, Sonia Rao, and Peptides Today. Before we sign off, a quick legal disclaimer. This podcast is for informational purposes only and is not medical advice. All compounds discussed are for research use only and not for human consumption. Always consult a licensed healthcare provider for medical needs and treatment.